Alright, ladies and gentlemen, here we are with lipids. So let's get started looking at their examples of lipids. If you look at the pictures on the first slide here, we have some oil. We have canola oil, olive oil, then we have butter. Again, here's a nice little picture of sizzling butter. Things that contain fat as well are things like some meats contain a fair amount of fat. Things that are cooked in fat, like French fries. And then we have things that are just totally made up of fat and fried would be your lovely little donuts. So as always, let's take a peek at our objectives and see what is going to be learned in this unit. So we have to, as always, describe the function of lipids, be able to give examples of fatty acids, phospholipids, triglycerides, steroids, and waxes, explain the hydrophobic and hydrophilic nature of lipids, and you have to know what those two words mean as well, differentiate between saturated and unsaturated fats, describe and draw the structure of a fatty acid, phospholipid, triglyceride, steroid, and wax, and then give the relationship of all of those. So let's begin with the function. Lipids give us long-term energy storage. And the reason they give us long-term energy storage is if you look at the diagram here, we have a picture of a lipid. And what do you notice? It starts out with an OH, double bond to an O, and then carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, carbon, hydrogen. This is what we call a long chain of hydrocarbons, and in those bonds contain a lot of energy. So we can get a lot of energy when we remove those bonds or break those bonds. The elements that make up lipids are carbon, hydrogen, and oxygen. The same as carbohydrates, except you're going to notice a big difference at the ratio in which they occur. And so if you look at our examples of lipids on this page, we have carbons and hydrogens, carbons and hydrogens, carbons and hydrogens, but oh, lo and behold, hardly any O's. So the way when you look at your molecules and you're trying to distinguish whether it is a lipid or a carbohydrate is, look, how many O's are there? And then again, what is the shape? These are in nice long lines, whereas if you remember, carbohydrates are in hexagons. And again, they're going to exhibit our nice little hydrocarbon chains. The monomers for lipids are fatty acids. The example we're going to be talking about is glycerol. It's a very nice long hydrocarbon chain with a carboxyl group. It has a COOH portion to it if you look at this molecule down here. And this molecule on this end is considered polar, which means it's attracted to water, which means it's hydrophilic. Hydrophilic means hydro meaning water, philic means like, so it likes water, or it's considered water-loving. It means it generally dissolves in water. Whereas the rest of the end, or the tail end of this molecule here, this long hydrocarbon chain is nonpolar. And remember, nonpolar doesn't dissolve in water, so we call that hydrophobic. Again, hydro meaning water, phobic meaning you're afraid of something or not liking water. So this fatty acid is a unique molecule in that it has one end of it that likes water and the other end that doesn't like water. When we look at our fatty acids, you're also going to have to look at them in the sense that are they fully straight lines or are they slightly bent lines? And that's going to indicate whether it's a saturated or unsaturated fat. And what does that mean for the makeup of the molecule? Basically, saturated fats are all carbons to carbons with single bonds. And that's going to make them, because you can't fit another bond in there because you've got your carbon to carbons and full of hydrogens, it's going to create an illusion or a picture that it's in a straight line. And all saturated fats tend to be solid at room temperature. So your things like butter would be a saturated fat. And they tend really not to be that good for you because they are actually a solid at room temperature. Your unsaturated fats generally have at least a carbon with a double bond, and that double bond, as you can see up here, is going to cause that little bend in the structure. So that's going to make them liquid at room temperature. And unsaturated fats tend to be much better for you than saturated fats. And the example of that would be like all of your oils, because they're liquid at room temperature. If we talk about our polymers of lipids, we have 4. The first one is going to be triglyceride, which is basically 3 fatty acids joined to one molecule of a glyceride, and hence the name tri, because there's 3 of them. Then there's phospholipids, which is simply just 2 fatty acids. There's waxes, which are long chains of fatty acids that have a little bit of an alcohol section in them. And then the last one is steroids. And they're going to be unique because you're going to look at their structure because their carbon rings are fused together. So we're going to look at each one of these on a separate slide. So let's take glycerol first. So if you look at glycerol, or triglyceride, excuse me, it's going to start with a glycerol molecule and be joined by 3 separate fatty acids. And so if you look at this one, it's got a long chain of hydrocarbons, another long chain of hydrocarbons, another long chain of hydrocarbons. These are all straight. There's no double bonds in here, so you know that would be a saturated fat. If you look at this diagram over here, you've got saturated, saturated, and an unsaturated. So oftentimes you can have a combination of your triglycerides, including a saturated fat and an unsaturated fat. Most of the foods we eat today are triglycerides, and that's why they contain a lot of fat that we could store as energy. And so we would eat a lot of these, and we utilize them when we can, but if we have extras, we're going to store them in certain locations as humans. And so females tend to store them on their hips, thighs, and maybe buttock area, where on males they tend to store extra fats in their gut. If we look at phospholipids, they're basically just 2 fatty acids. So if you look at this diagram here, you have the polar end and the 2 strands of fatty acids attached here. Now, remember where we said that we're going to have to be able to draw and depict these. So for a phospholipid, you're going to depict the top part with a circle, and then the tails or the ends, the fatty acids, as little squiggly lines. So it's kind of like a balloon with 2 lines to it. Because these 2 are squiggly, these would be unsaturated fats. If I put straight lines, they would be saturated fats. So you can depict them as either saturated or unsaturated. And phospholipids are mainly contained in cell membranes. So if you look down at this diagram, it's a complex drawing of a cell membrane. And so here you have your one set of balloons, your other set of balloons. So there's your 2 fatty acids that make up cell membranes. And cell membranes are going to be something that we're going to spend a considerable amount of studying later on in the year. Waxes, you guys know beeswax, candle wax, they're all kind of familiar with something like that, but you also maybe don't look at leaves that often, but a lot of leaves have a wax coating on them. And basically all you really need to know for waxes is that they're waterproof and they're also protective layers. So if you think of a plant that has a waxy coating on it, it's really hard for an insect to chomp and chew on it. And then waxes are also waterproof, so they repel water. So like feathers might have a waxy coating, and then as we said, part of a lot of substances need to be waterproof to protect from water getting in, so they would be made up of a wax. The last group, and everybody always forgets or doesn't think that steroids are part of the lipids, but they are. And you guys probably think of steroids as the things that you see in TV or something when people are making massive amounts of muscle and having steroid shots and illegally using them from the baseball scandal and so forth. But basically they include hormones which are going to control a lot that goes on in your body chemically. Things like estrogen, it's going to make females female, and testosterone, which makes males males. And then the other big steroid that you don't really think of as a steroid, but that's cholesterol. And maybe some of you have heard your parents or your grandparents discussing high cholesterol levels that they have. And cholesterol is a substance that sticks to your vessels, and it can clog your arteries and cause heart attacks and all sorts of different things. So you have to really be careful how much cholesterol you take in. And the interesting thing about steroids is how could you recognize them is that they have this unique structure here where the hexagons are fused. You notice there's not a line indicating a covalent bond between the two, and that's how you would distinguish the difference between a carbohydrate and a steroid, is that the hexagons are fused in a steroid and they're not fused in a carbohydrate. So let's look at and kind of refresh our memory on how do we make these complex molecules. And if you remember, we're going to use the chemical reaction of condensation, or also known as dehydration. And so you're going to start with a glycerol, and you're going to add fatty acids to it. So if you look at this glycerol, remember we're going to remove an H and then an OH from here, and then you're going to create your water molecule. So if I was trying to make this triglyceride, I would end up removing one H and one OH from each of these and creating 3 water molecules. And remember, this is all governed by enzymes coming in and helping this process take place. It wouldn't occur very fast if we didn't have our enzymes, and it would require a great deal of energy if we didn't have our enzymes. how do we break our triglycerides down so that we can get energy is, again, we're going to use the process of hydrolysis. So we're going to take our nice little triglyceride here, add in some water and our enzyme, in this case lipase, and we will break it down to a dry glyceride and get some energy and remove one of the fatty acids. Put in some more water, some more enzymes, and I break it down into a monoglyceride, and I have my fatty acid. So again, this is how we take in our foods that are high in fat content, our triglycerides that we eat, break them down and get energy. That pretty much does it for lipids. So let's look at your review. Can you give me an example of lipids? Do you know the function, the elements that are involved, their shape, the monomers, whether it's a saturated or unsaturated fat? Is it hydrophilic or hydrophobic? And what are the 4 polymers that exist? And then how do we make and break our lipids? That should do it. Thanks.